Okay. Lakshmi, how are you? Am I audible and visible? Okay. Am I audible, visible?
Yes, ma'am.
Okay. Your health is now okay? Okay.
Ma'am, you did not send yesterday's PPT.
Oh, I have not sent.
Yes.
Oh, sorry. I will share it, okay?
Okay, ma'am.
And I have shared some videos too. You
Yes,
have
ma'am.
to observe that. है ना?
Okay, ma'am.
You have to watch those videos. So related to that plant vector and few question answers related to recombinant DNA technology. Okay?
Okay, ma'am.
Acha. So today I'm going to discuss about immunoprecipitation. This is the next topic. Whether should I go for this immunoprecipitation or you want to understand that immunoblotting? Not that immunoblotting, that southern blotting, northern blotting, and western blotting. Already I have discussed. I think from that discussion you understood the topic. What do you think?
We did not complete yesterday, ma'am.
So you want to continue that?
Yes, ma'am.
Okay, no problem. Let's just a minute. Second. Okay, so how blotting a lot of background music.
Yes, ma'am. I just.
Okay, just see. Uh, is it visible? Just a minute. No, it's not changed. Okay, just a minute. I have to change the topic. Precipitation. I have to change it. No problem. See, Lakshmi. Uh, After this immunoprecipitation class, I will continue that topic. Okay? Is it
Okay,
okay
ma'am.
for you? है ना?
Fine.
Changing is very difficult. Okay. So the topic is immunoprecipitation. So from the name you can understand immuno plus precipitation. Immune means if you consider any immunological assay, so most of the cases, in that case, antigen involved, antibody involved. Okay, and there should be antigen and antibody interaction. In ELISA also we have observed those things. In case of, if you consider ki Western blotting, in that case also you can use antigen antibody interaction. Now, if you consider the immunoprecipitation technique, so in immunoprecipitation technique, we generally identify antigen molecule in a mixture. Okay, if you consider ki in a mixture, a lot of antigen molecules may be possible. Like if A present, then B present, then C present, D present. Okay, so multiple antigen, if in a mixture multiple antigens are present, so in that case what will happen, if you want to identify only one antigen, how to identify that? If you add specific antibody, okay, specific antibody against this antigen, what will happen? This antibody only bind with the, it will bind only with specific A. Okay, because we are adding specific antibody against A. In this way, what is happening here? Whenever you are adding specific antibody, antigen antibody interaction will be there, binding will be there. So finally, what will happen? This A will be precipitated with the specific antibody. Okay, from the mixture. Okay, so A, B, and C, if you consider these are multiple antigens present, we need only A antigen. So, in order to purify that, or you have to separate the antigen or identify the antigen, you have to you can precipitate that antigen if you add antibodies against that antigen. So that is the main thing in immunoprecipitation process. That means antigen binding with antibody, specific antibody. Okay. So now, first of all, let's see what is happening here. Okay. Okay. So in this case, first of all, see what is the definition of immunoprecipitation. Immunoprecipitation is the technique of precipitation or precipitating a protein antigen out of solution using an antibody that specifically binds to that particular protein. Whatever I told you in my previous slide, exactly it is given here. If in a mixture, if you have multiple types of antigen in the mixture, if you want to separate your antigen of interest, you have to add antibody against that antigen. Okay. So what will happen? This antibody will bind with the specific antigen and there will be precipitation. Okay. Here I have given only two types of antigen present, blue and red color. Against red only antibodies are there. So you want to precipitate antigen, red colored antigen. So you have antibody against that. You just add an antibody and what will happen? Specific antigen will bind with the this antibody and it will be separated. Okay. Now this process can be used to isolate, concentrate a particular protein from a sample containing many thousands of different proteins. So this is happening here. We are isolating your protein of interest from a mixture. So that means you are doing the concentration. You are concentrating the sample. Okay, and whenever if you know about the protein precipitation technique, you know ammonium sulfate precipitation is done, and if you add acetone in the media, if protein is present, acetone can precipitate protein, alcohol can precipitate protein. Okay, so in that case, we do precipitation process in order to concentrate the sample. How? If you consider here, initially 100 ml sample was there, mixture was there, 100 ml. Okay, in 100 ml mixture, few protein particles are there. So I'm considering about the protein precipitation. So in that case, it will be finally if you add the ammonium sulfate or alcohol, what will happen? Only the protein molecules, not other molecules. Only the protein molecules will be precipitated. Okay. So now you just dissolve this precipitated sample into very small amount of liquid. You can mix it with only 10 ml. So what will happen? The volume is completely reduced, right? So from 100 ml it is reduced to 10 ml. That means, and already you are precipitating the only sample, your protein of interest, not the carbohydrate, not DNA, nothing. So and you are dissolving it in very small amount of liquid. So what will happen? This is the we are doing the concentration, concentrating the sample, है ना? In this way, in immunoprecipitation also, if you consider these are the antigen, red color antigen. Initially, it was in 100 ml solution, okay, 100 ml blood or whatever you consider in the buffer may be possible. So, if you add antibody inside it, what will happen? Antibody will bind with those specific antigen, and there will be precipitation, and you can make it in a concentrated form. Okay, so we are doing separation as well as we are doing the concentration, concentrating the sample. Okay, na? So here it is written isolated, you are isolating and concentrating from the mixture. Okay, so this is the main purpose for the immunoprecipitation. Immunoprecipitation requires that the antibody be coupled to a solid substrate at some point in the procedure. In this case, not only antigen antibody interaction is happening, in this case, one new thing is there that is called the mainly agarose beads, or it is magnetic beads. Okay, why those are used? In order to in order to separate the precipitated sample very easily, okay, beads are added. Beads means either it is magnetic bead or it is agarose bead. So in this process, what is happening? What is happening? This picture is visible to you. Is it visible to you?
Yes, ma'am.
This see the picture. This is antigen mixture. Okay, lot of antigens are there. You need only green
Which
color.
slide you're in? I'm still in two.
Huh?
Which slide you're in?
Oh,
Because I'm still in two.
this is, I think, रुको, wait. This is two. This is two only.
Okay, ma'am. Then visible.
You just focus on the right side picture,
Okay, ma'am.
not whatever I have I drawn. Don't see this. I'm removing this one so that you can understand it. What is it? Okay. So if you see here now, I'm trying to increase the volume structure of this part. Okay, try. Okay. So now see what is happening here. In this case, a lot of antigen mixture, a lot of antigens are present. See, blue color, dark blue, light blue, green, and three types of antigens are present. From this mixture you want to separate green colored antigen. Okay, so you have to add primary antibody. Primary antibody, I told you during the discussion of ELISA, what is primary antibody? If it one antigen is present against antigen, if you are preparing antibody, that is called primary antibody. Now what is secondary antibody? Secondary antibody means it will bind with
Primary
primary
antibody.
antibody, है ना? So initially in this case you are adding primary antibody. Primary antibody will bind with the actual antigen. Now in the next step you have to add the secondary antibody, but or secondary antibody or protein A or protein G. Okay, and secondary antibody is actually attached with agarose beads or magnetic beads. It is written here, agarose beads or magnetic beads. If magnetic bead is there, you can separate the precipitate very easily, and as well as for the agarose beads also. Okay, so here what three things are there: antigen, then primary antibody, then secondary antibody. But fourth thing is, with secondary antibody, what is attached? Either agarose beads or magnetic beads. See here, if you consider key with this if you consider this is one magnetic bead, okay, bigger size magnetic bead. With magnetic bead, what is attached? Actually, secondary antibody, है ना? Now, with secondary antibody, what is attached? Primary antibody, है ना? Primary antibody is attached. Now, if you consider it is only attached with the green color antigen, so antigen will be at this position. Clear? So, in this way, with one magnetic bead, a lot of secondary antibody will be there. See, in the surface of the bead, a lot of secondary antibody is there. है ना?
Ma'am, can you zoom the picture?
It is not visible. Just
It's
a
very
minute.
small.
Okay, I'm trying to make it separate.
And ma'am, I think your video is freeze.
Again, अरे भाई क्या करें? Now is it visible? Hello?
No, ma'am, it's freeze.
This is also freeze. Now,
Yes, still freeze in slide 2 and there is no movement. Even your video is freeze.
क्या करे बोलो? They are not allowing me in Jeep.
Ma'am, is your
Giving
network fine? Is your network
my
good?
no here network is quite good. There is no problem.
I think my network is also good.
Then why it is happening?
Okay, ma'am. Ma'am, shall I leave the class and join again?
Okay, you try it.
Okay, ma'am.
See, is it visible?
Yes,
Now it is visible.
yes,
Primary antibody, secondary antibody, beads, everything visible.
yes, ma'am.
Okay. So now see what is happening here. Antigen mixture. In the second picture, it is primary antibodies binding with specific antigen, green colored antigen. Okay. And the third step, secondary antibody is given. Secondary antibody is actually attached with magnetic bead. Okay. So this is actually a magnetic bead or agarose bead. So with one agarose bead or magnetic bead, a lot of secondary antibody can bind. Okay. So this is happening. Now, if you wash it, okay, if you wash this mixture, what will happen? The unwanted antigen will be removed. So blue, dark blue and light blue colored antigen will be removed. And in the solution, what will be present? Only magnetic bead, secondary antibody, primary antibody and antigen. Okay, your interest of antigen. Now you just elute. Elute means in this case you have to separate, na? Because what is the purpose? Why? What we are trying to do here? In immunoprecipitation, what is the purpose of doing the immunoprecipitation? Tell me why you are doing such things. We are trying to isolate our antigen, na?
Antigen of interest, ma'am.
Antigen of interest. So in order to do that, you have to separate from the immunoglobulin. Antigen should be separated from the immunoglobulin in the final step. So that is happening during the elution process. And whenever you are doing this SDS page of this mixture, all the bands will be, you will give separate bands you will get for primary antibody, secondary antibody plus your antigen of interest. Okay, so at least you will get 3 bands in the SDS page. Now if you consider, you only need the band for antigen A. Okay, so for antigen A, if you consider this is the, you can't understand from the
Actually, the
SDS
video is
page which
freeze
bond
again.
is, huh?
The video is freeze.
I'm giving only same picture, same slide.
Okay, but your video, like, ma'am, is like freeze.
What can I do? Every time it is happening. There was no such problem during the Google Meet. Hannah? It was quite good. Just
Okay, ma'am, you can continue. We'll see if uh,
okay.
fixes by itself.
Okay, so now see what is happening here. But you don't know na which band is for your antigen of interest. है ना? So in order to identify the band, you have to do what? You have to do in the next step, you have to do the Western blot. Okay. So Western blot, I told you about the Western blotting process. Oh, I have not discussed the Western blot. But you just understand कि in Western blot process, what is happening? HDS page, these bands are transferred to one membrane. Okay, nitrocellulose membrane. It's like filter paper, just paper. On the surface of paper, all the bands are transferred, and then what is added? Antibody against the antigen, specific antigen, it is given. And that antibody is actually attached with one probe. Means probe is the antigen, but antigen is attached with mainly radioactive or enzyme linked. Okay. So in that case, what will happen? What will happen? That antibody will bind with the antigen A, and there will be only one band. These, after Western blotting, you can't see these two bands. Only this band will be visible. So you can this is the confirmatory test. Key, your antigen of interest is present and you have isolated it. You have separated it. Clear? Is it clear? Hello?
Now, which band will be visible? Like only one band, or that will correspond
After,
to?
only one band. Western blot, after Western blot, it is, you have transferred the all the three bands into Western blot. Western blot means mainly membrane is there. Okay, nitrocellulose membrane. On the surface of membrane, three proteins will be transferred. Okay, so initially three proteins will be transferred, exact copy of the SDS page on the surface of nitrocellulose. You are making the stamp. You are taking one stamp from SDS page, and it is, you are giving on nitrocellulose paper. So what will happen? Initially there will be transfer of three bands. Okay, but it will not be visible because we are not giving any stain or other thing on the nitrocellulose paper. Okay, when it will be visible? If you add specific antibody against your antigen of interest. So what will happen now? In the next step, what will happen? There will be, like, if you consider key, this is the SDS page. I'm just again drawing it. This is three bands. Okay, these are three bands. You need only this one. If you consider, suppose you know key, this is the band. Now you have to transfer this band onto nitrocellulose paper. Okay, on the nitrocellulose paper, there initially there will be these three bands, but it not be visible. Okay, but in SDS space it is visible because you are adding Kumasi blue not to visualize the bands. So now you have transferred these three proteins on the surface of nitrocellulose membrane. Okay. Now what is happening? Next step, in the next step, this is one part of the Western blotting. What is happening? On the surface of this nitrocellulose paper, you have to add, what you have to add? You have to add antibody. Okay, and this antibody is actually attached with some label. Okay, and this antibody is against your protein of interest or antigen of interest. So what will happen? This antibody only it will bind with your protein of interest, है ना? So this antibody will bind with only this band. Now whenever you check and wash everything, what will happen? You will see only one band here, because antibodies are not binding with other two bands, other two proteins, only with the antigen of interest. And in the next side by side, you should use some control. Okay, so already you have antigen of interest and antibody of interest you are adding now in the nitrocellulose membrane. So there will be control, there will be one band already. So you can compare कि exactly same thing is happening here. है ना? It's like in the SDS space, we generally run standards in order to determine the molecular weight. है ना. Different mixture of standards are given with known molecular weight. That is, from that you make one standard curve, and from the standard curve we generally measure the unknown protein molecular weight. In this case, everywhere, whenever you are doing any test, you should have some reference or standard. So in this case also, during whenever you are doing the Western blot or anything, there should be one lane where you are adding the control or reference, so that you can exactly match with that result. Clear? This is actually you are running reference. In the reference, what is present? Only antigen of interest is present, not three other two immunoglobulins. Only antigen of interest is present. But in the test lane, actually three bands are there. Initially, three bands are there, but you are adding antibody, so only one band will be visible. Clear? So this antibody is actually binding with your test sample as well as with your reference, है ना. So therefore you are getting the exact band side by side in the reference as well as in the test sample. So confirmatory, this is the confirmatory case that your protein of interest is present. In this way, what is happening? Immunoblotting, immunoprecipitation is done. So initially what is happening? In the mixture, you are adding antibody, and that antibody is primary antibody. Now add secondary antibody. That secondary antibody is actually attached with agarose beads or magnetic beads. Okay. Now you just wash it. What will happen? Unbound antigen will be removed. Only your antigen of interest will be attached with the primary antibody. And now you elute it, means separate all the three proteins through SDS page, and then do Western blotting for the confirmatory test. So these are the actual sequences uh, uh, during the method of immunoprecipitation process. Okay, what do you think? Is it clear?
Yes, ma'am.
Oh, now see. In this case, mainly, it's like ELISA test, direct method, indirect method was there. In this case also, direct method and indirect capture method is there. So direct capture means you are not adding any secondary antibody. Only primary antibody, with primary antibody beads are there. Okay. But if you consider about the indirect method, whatever we have discussed, this is actually indirect method. Because here, in case of ELISA also, if you remember, in indirect method, secondary antibody was labeled with enzyme. Okay. Can you remember it? In direct method, only antigen antibody interaction was there. But in indirect method, interaction was with antigen, primary antibody, secondary antibody. है ना? So this method is exactly like indirect method is like your ELISA indirect method. So this one is actually indirect method. But if you just add beads here in the primary antibody, so there will be this step. There will be no, this step will not be there in the direct method. Secondary antibody is not involved in that case. Okay. So directly from primary antibody, you can go to this step. Okay. There will be no secondary antibody involvement. Clear? So direct, indirect, exactly like ELISA, nothing else. Direct means always remember only antigen, only antibody involved. Only antibody means primary antibody actually against the antigen. Okay. So this is happening. Now, if you consider about the steps of immunoprecipitation, how it is done, already I have given, but to some extent you can start with lysis of the cell. So from the cell you have to collect antigen. So say lyse cells and prepare sample for immunoprecipitation. Second step is the pre-clear the sample by passing the sample over beads alone or bound to an irrelevant antibody to soak up any proteins that non-specifically bind to immunoprecipitant components. Third step is incubate solution with antibody against the protein of interest. Antibody can be attached to solid support Solid support means beads. Before this step, direct method, or after this step, that is called the indirect method. Now continue the incubation to allow antigen antibody complex to form, and finally you get precipitate the complex of interest and remove it from the bulk solution. And now you can do washing to remove the unwanted antigen. And finally what you have to do, the SDS page, then western blotting. So this is SDS page, and analyze the complex of antigen of interest. This can be done a variety of way. Western blot you can do multi mass spectrophotometric determination you can do, or you can do through ELISA test. Okay. Through that, those are confirmatory tests. Now uh, see, this is analysis. How analysis is done? Analyze complexes or antigens of interest. This can be done in a variety of ways. It may be done through SDS page followed by gel staining. Now in the next step, what you have to do? HDS page followed by gel staining, cutting out individual stained protein bands and sequencing the proteins in the bands by multi-mass spectrophotometry. What is that is meaning? What is they are trying to tell you? That is, whenever, if you consider this is the gel, okay, this is gel. Now you have protein of interest and you have two other bands, है ना? Two other bands. Okay. Now generally multi-mass spectrophotometry, okay, mass spectrophotometry, multi-mass spectrophotometry. So this is very sensitive method, or this instrument is very costly. Okay, but you can analyze any sample. It will give you molecular mass, and their database is there. There will be comparison between the database and your sample, and result will they will give you the result. This sample is for this molecule. Okay, in this way, database is there. So in order to do that, generally what is happening, from the HDS page, separate bands are actually separated. That means what I am trying to tell you, that is, if you consider these three bands are there, okay, you have to cut every band. In this way, you have to cut every band. So you make three separate sample, cut all the bands from the gel and dissolve it. And there is method how to dissolve the gel and how to prepare monthly sample. Okay, so main thing is you have to separate all the bands by cutting. Just manually you cut all the bands whenever after the staining. Okay, three bands you can separate it from three separate sample, है ना? Now you just load these three bands in the multi instrument. You will get different mass for different sample, and comparative data is there, I told you. So comparative study, they will tell you that if your interest, protein of interest is present here in sample A, if you consider this is A, B and C, okay, these are three bands and three samples are there for Marley. Now you can compare all the molecular weight, and from the database you can identify that this A is actually for your antigen of interest. Clear? So that is the confirmatory test. It's like Western. You are not doing Western blotting because you don't have antibody against that antigen. It may be possible, which is radio labeled or anything. So if that is not there, if in your laboratory multi-mass spectrophotometry is there, you can do in this way. You just cut all the bands and prepare the sample for multi, load it and analyze the data. Okay, confirmatory test. Now if or you can do Western blot. If radioactive labeled antibody is there in your lab, you can easily do it, Western blotting. Clear?
Yes, ma'am.
Huh. So either radioactive or chemiluminescent probe is attached, or fluorescent compound is attached. Okay, generally, whatever I told you, labeling of any protein. If this is one protein, labeling is done in immunoassay. That means ELISA, enzyme linked immunoassay. In that case, enzyme was labeled, and an enzyme was attached. But in some cases, there may be attachment of chemiluminescent molecule will be attached in order to visualize the sample, or maybe fluorescent sample is attached, fluorescent compound may be attached with this protein, so that you can measure it through the fluorescent fluorometer, yeah, fluorescent spectrophotometry, no, fluorescent fluorometric method. Through the fluorometric method you can identify fluorescent compound. Okay, so these are identification process. Radioactive, maybe radioactive compound is attached, or chemiluminescent compound is attached, or fluorescent compound is attached, or enzyme may be attached. Okay, so in this way you can identify your sample. Clear? Okay. Now next, in this table what is given, monoclonal and polyclonal antibodies given. So most of the cases we use monoclonal antibody, because in the mixture specific antigen is there, now one type of antigen is there. So against that specific antigen, if you use monoclonal antibody, it will be better. है ना? Monoclonal means only identifying only one type of epitope. You know na difference between monoclonal and polyclonal. I told you before the ELISA discussion. You know or not?
I know, ma'am.
That's very good. Chalo. Now, in this case, comparison beads. Several beads can be utilized. Na? Agarose, then magnetic bead, or combination. Magnetic bead plus agarose may be added. So comparative study is given. Whatever in your laboratory it is available, you just use. Magnetic bead is better, I think, as compared to agarose, because if you wash it, na agarose, there will be um, loss of the sample. Okay, so sample loss may occur during the washing step. Magnetic bead is not so much. It's good. Sample loss is negligible. See, so if you combine agarose plus beads, it will be much better. Everything is there. You can do it. Okay. Now application where it is applied. Main purpose is to isolate the antigen protein. Okay. So here application C, isolate or detect the protein, concentrate the protein, enrichment of low abundant proteins. Now study protein-protein interaction. That is also done if you do the immunoprecipitation. Identify unknown proteins and verify the protein expression. After the protein expression, after the expression of the protein, you can verify whether protein is expressed or not through the immunoprecipitation technique. So mainly ELISA, Western blot, मेन पर्पस ऑलमोस्ट सेम है. ठीक है ना? बट यहाँ पे देखो प्रोटीन प्रोटीन इंटरेक्शन एक न्यू थिंग है. यू कैन डू फ्रॉम द इम्यूनोप्रेसिपिटेशन टेक्निक. So now going to in this from separate source, I am going to discuss about the different methods of immunoprecipitation. Okay. From this source, definition of immunoprecipitation is exactly same. See, immunoprecipitation is the technique of precipitating a protein antigen out of solution using an antibody that specifically binds to that particular protein. Okay. Now, this process can be used to isolate and concentrate a particular protein from a sample containing many thousands of different proteins. Clear? So this is the actual immunoprecipitation technique. Now, I'm going to discuss about the different methodology. Sorry. Here, first of all, I am going to discuss what are the methodologies used in immunoprecipitation. Mainly two methodologies are there. In one sequence, that means antibody against the target is pre-immobilized. That means in the first method, what is sequence is slightly different. These two methods are actually sequence are different. That means whether antigen you are adding first or antibody you are adding first, that is the difference, nothing else. So in one sequence, the antibody against the target is pre-immobilized. So that means first of all you are adding antibodies. So in a container, if you consider, you just add antibody first. Okay, first of all you have to add antibodies, specific antibodies. Okay, now add the mixture of antigen. Okay, so this is the sequence. See, target is pre-immobilized onto an insoluble support such as agarose or magnetic beads and then incubated with a cell lysate containing the target protein. Clear? This is actually direct method completely. Now, if you consider alternatively, free antibody is allowed to form immunocomplexes in the lysate and then the complexes are retrieved by the beads. So, and then another process is, first of all, this complex formation is done, and lastly, it is separated through the beads. Okay. Here on first of all, agarose or beads are attached with the antibody, and then you are adding antigen. And in this case, antigen antibody interaction is done. Then you are through the beads you are retrieving it. Okay. Now, if you consider the method, what are the different processes of immunoprecipitation? Okay. First of all, individual protein immunoprecipitation. So, individual protein immunoprecipitation is exactly whatever we have discussed till now. Okay, that means antigen, then add primary antibody, then secondary antibody which is attached with the beads. Secondary antibody you can use or protein A and G. Okay, so this is actually individual protein immunoprecipitation, actual normal one, normal immunoprecipitation. Now, second method is the co-immunoprecipitation. Co-immunoprecipitation means, co, from the co you can understand, co means together, है ना? So in this case, if you consider, in this process actually we did understand protein-protein interaction. Whether two proteins are attached together or not, we can check it through the co-immunoprecipitation technique. Okay, so in this case, if you consider key, you have A antigen and B antigen, but A and B both are attached together. Okay, you want to study whether A and B attached or not. That is the protein-protein interaction. Na? You have to check it. You have to check whether they are attached together or not. In order to check the protein-protein interaction. So through the co-precipitation, you can understand it. You can determine that A and B, they are actually present. They are actually attached together. How it is possible? First of all, you have only antibody against A antigen. You don't have antibody against B antigen. Okay. So first of all, you have to add antibody against antigen A. Then what will happen? If A is attached with B, they precipitate together. है ना? Whenever you are only adding antibody against A, if A is attached with B, what will happen? Immunoprecipitation will be done for A and B as well as for the antibody against A. Clear? Is it clear, beta?
Um,
है ना? In the precipitation, you are not only getting A and antibody. You are getting A plus B plus antibody against A. है ना? So therefore, this process is known as the co-immunoprecipitation. So A and B together precipitation is happening. So it is known as immunoco co-immunoprecipitation. Okay, in this process we understand, analyze the protein-protein interaction. Like if you consider ligand receptor interaction, okay, if you're checking, you have antibody against ligand, you can check whether receptor is there or not. Okay, in this way, or cofactor enzyme interaction, you can understand it through this immunoprecipitation technique. Okay, the basic co-immunoprecipitation protocol is exactly same. Okay, exactly same like whatever I told you previously, exactly same. But finally what will happen, I will discuss that. So this is the basic co-IP protocol is the same as that described for IP, and indeed any system designed for IP should also work for co-IP. So if IP system is there in your lab, you can make it to co-IP. Now there are a number of additional factors to be considered. So a lot of things are also there, especially for the co-immunoprecipitation technique. So those are few things are also there in immunoprecipitation technique. Like you have to check all the medium, all the buffer, washing technique. So in every step, you have to be cautious during the process. Okay, so here it is given the optimization of binding and wash condition must include consideration of effects on bed prey interaction as well as on that between antibody and bed. Bed prey interaction, what is bed prey interaction? Bed prey interaction, maybe just a minute. Bed prey interaction. I don't know what is the meaning of this. BAIT and PREY. Na? Two hybrid acid checking for interaction between two proteins called here bait and prey. Achha. This is protein-protein interaction, nothing else. Hybrid system. No, so you don't have to think about it. You just thinking what is happening. What is the difference between co-immunoprecipitation and normal immunoprecipitation? So in this case, you just I'm removing everything. So much of color is there. See. In this case, what is happening? We are checking antigen protein-protein interaction. So in that case, if you see in this figure, okay, green color protein and red color protein, they are attached together. Clear? From this picture, green color
Yes,
and
ma'am.
red color, they are attached together. You just consider ki green and A, they are A and B. Okay, so this is A and B, they are attached together. So you have antibody against A. Okay, you have antibody against A only. So what will happen? A and B both will be precipitated. Okay, and you just precipitate it, spin it, centrifuge it. What will happen? All the antigen antibody complex will be precipitated. Now you just do the SDS phase. What will happen? You will get three types of band, है ना? In this case antibody is attached with one protein, one probe. Okay, so or a beads, magnetic bead may be there. So in that case, what will happen? Finally, after the SDS phase, you will get three bands, two bands for two protein and another band for the antibody. Clear? Is it clear up to this?
Yes, ma'am.
Okay. Now in the next step, what you have to do? Now you have to add antibody. No, you have to do the Western blot. Okay. So in the Western blot, you are just transferring all the three bands. Initially, you are transferring all the three bands. Okay. Now you just add on the nitrocellulose paper, you have to add antibody against the green color, or green color means antigen B. Okay. So you are adding antibody against B. Initially, you are adding antibody against A, but in the next step, whenever you are doing the Western blotting, at that time you have to add antibody against second protein. Okay. So what will happen? that second antibody will bind with the second protein, है ना? So therefore you will get only one band. If you are getting one band, not other two bands, so you can confirm that this B is present in the sample. B was attached with the A. Okay, then only you are getting this type of bands. And simultaneously, I told you na ki always reference should be there. So B antigen, known B antigen will be there, so that antibody against that B antigen can bind. Okay, so what is happening here? Again, I am trying to just summarize it. In the
Ma'am,
mixture,
only the last step. Yeah, you can repeat this one. We'll
this
transfer it to nitrocellulose membrane, then
then you just add antibody against the second
specific for
protein.
the
Second protein, whether that second protein was attached with A or not. So if you consider initially it was A, antibody against A, now you are adding antibody against B in the nitrocellulose. Initially, during the precipitation process, you are adding
okay, during the
antibody. Uh,
first precipitation, we are adding antibody against A. Then in the last step, during nitrocell after nitrocell transferring on the nitrocellulose membrane, we're adding antibody against B.
huh. You are adding antibody against B. Then only you can confirm na ki B is coming together with the A. Then only precipitation is done. Because initially you are not adding antibody against B, but it is coming. Why it is coming? Because they are precipitated together.
Okay, ma'am.
So that is the thing. Reference is, I am always talking about reference because everywhere you will see reference is there, reference is there. So yeah, I am informed. Okay. So reference is just for the confirmatory key. This exactly same position band is coming. Okay. Nothing else. So this is called the immunoco-precipitation. Clear? So in this case, same thing is happening. Only few simple two steps you can consider ki different hai. ठीक है? Nothing else. अच्छा. Now next process is the chromatin immunoprecipitation. Chromatin immunoprecipitation or CHIP. Try to remember the short form. CHIP. Chromatin. Chromatin. What is chromatin? Chromatin means DNA. Okay. So double stranded DNA plus few proteins, especially histone proteins or any other protein. Well, lot of protein can bind with DNA, na? DNA binding protein. So like transcription factor. Transcription factors are attached with the DNA. So those are actually protein, na? So different transcription factors or any DNA binding protein and histone protein. Okay. So in this process, we understand or you can sequence the part of binding of the protein with the DNA. That means DNA binding domain you can identify. Yeah, here we are not identifying the protein. Mainly we are, you can do the same thing. Protein also can be identified, which protein is attached with the sequence. But mainly we identify the sequence where protein is attached. If you consider the transcription factor 1, where in which position of the DNA it is attached, you can sequence it or you can identify it through this CHIP method. Okay. So see, just read. CHIP is a type of immunoprecipitation used to investigate regions of genome associated with a target DNA binding protein. Is it clear? So at the region, everywhere proteins are not binding with the DNA. है ना? There should be specific region. So if you want to identify which region this protein is binding, you can identify through chromatin immunoprecipitation technique. Clear? So such as transcription factors, histones, or conversely to identify specific proteins associated with a particular region of the genome, and it is commonly used in epigenetic study. Okay, so this is the main thing happening. Now, what are the steps involved in this case? First of all, growing and collection of material. You just collect the sample you want to analyze it. So the next step, what you have to do? Cross linking. Cross linking means cross linking between protein and DNA. So in the solution, DNA protein binding is happening in the sample, but though that binding is not strong. So in order to do the cheap method, you have to make the bonding very strong between protein and DNA. In order to do that, you have to do the cross linking. Cross linking means you have to add formaldehyde. If you add formaldehyde, what will happen? It will fix protein and DNA together. Okay, so we are studying what we are studying here, protein and DNA interaction. In the previous method, in the co-precipitation technique, we are studying protein-protein interaction. Here we are studying actually DNA-protein interaction. Okay, so in that case, protein and DNA, so proteins glued to DNA by adding formaldehyde. Okay, so all the proteins, whatever unwanted protein, your protein of interest, everyone is attached with the DNA. Now next is chromatin isolation. Okay, so in that case, fragmentation is done. Oh, no, first of all, from the cell, you have to separate the chromatin. That is called the chromatin isolation. Next step is the chromatin fragmentation. If you do the fragment, what will happen? Different fragment, you will find different protein attached with that. So in this figure, see yellow colored protein is attached with one fragment, pink color is attached with one fragment, and blue color is attached with another fragment. Okay. Now you want to identify which protein? You want to identify yellow color protein binding site. Okay. Now you know that you want to study this interaction. This yellow color protein, where it is binding, you want to know. So that means you want to sequence this part. You want to sequence this part. Okay, similar. It will be similar because yellow color protein is attached. So in that case, what will happen? You have to add antibody against this yellow color protein. Clear? So antibody which is attached with the beads. Clear? So in this case, what will happen? From the mixture, you are isolating your protein of interest. है ना? So isolation with antibody. Up to this, is it clear? Tell me.
Um,
है ना? So in this, so first of all, cross-linking, then chromatin isolation, then you have to do fragmentation of the chromatin and add antibody. Antibody against whatever you want to identify against that protein, you just isolate it. Isolation with antibody. And next step is you have to digest the protein because you are trying to sequence the DNA. You don't want protein. So in that case, you can digest that protein. Okay, so protein digestion, and finally you do the DNA purification. And after DNA purification, you do PCR analysis. So through the PCR, you can also sequence it or PCR analyze means through the PCR. Generally, we do amplification of the number and a copy number is increased, and you check the concentration and match with any similar type of DNA fragment with the control or reference. So you can and do the sequencing. Obviously, sequencing should be there so that you can identify the region. So in this way, which method we have discussed, this is actually CHIP, chromatin immunoprecipitation. Okay, chromatin immunoprecipitation. So through this process, you can identify the DNA part. Even you can identify the protein part also. है ना? In that case, you have to digest the DNA. Or if you do digest the DNA, you can get the protein and you just do the Western blotting after that if you want to identify the protein. है ना? That is also possible. DNA sequencing is possible as well as that protein identification is also possible. So in this way, you can study DNA and protein interaction. Now next is the RIP, that is RNA immunoprecipitation. In RNA immunoprecipitation, exactly just DNA precipitation Whatever done in the DNA precipitation, here also same thing. But in this case, it is RNA specific protein. Okay. But initially it was DNA binding protein. In this case, in chip, it was DNA binding protein. In case of RIP, we are studying RNA binding protein. Clear? So RIP is similar to chip, but RIP is performed using an antibody that targets a specific RNA binding protein. So initially in chip, in this process, in the chip method, we are targeting DNA binding protein. So against DNA binding protein, you have to develop the antibody, and that antibody is attached with the bait. Whereas in case of RIP, you are actually developing antibody against specific RNA binding protein, nothing else or nothing. Now RNA protein complexes are separated by RNA extraction exactly like DNA method, nothing else. Okay? And now finally you have to do the RT-PCR because it is the RNA. Now finally you are getting the RNA. In this case you are getting DNA. So simple PCR analysis you can do. But in case of RIP, finally you are getting the RNA sequence. So RNA always you have to do the RT-PCR, reverse transcriptase PCR. You can get the you can get cDNA and do the sequencing. Now just complementary sequences the RNA. If you do the sequences of cDNA, you can make the complementary sequence, and that is for the RNA. Clear? है ना, RNA separation clear. So in this case what is happening, so this is one example. If you consider during the spliceosome na splicing process, so in that case several RNA binding proteins are attached. You can identify that region where RNA binding proteins are attached through this process. Okay, splicing, you know na, exon intron removal, exon attachment, you know na, splicing process. That is for prokaryotic system or eukaryotic system?
Eukaryotic.
Always eukaryotic. In that process, hnRNA is converted to mRNA. है ना? And in that case, finally, poly A tailing is done, cap at the 5' end, that is done. But splicing means mainly intron removal and exon joining, and that is through the spliceosome complex. So in this way, you can separate the RNA fragment, and you can do the sequencing through RT-PCR and cDNA sequencing. Now another final method, that is the tagged protein IP. Tagged protein IP means if you don't have any antibody against your antigen of interest, you can use this thing, tagged protein IP. How? Due to the lack of an available antibody or a specific target, many proteins cannot be immunoprecipitated. है ना? Main thing in immunoprecipitation is you should have antibody against that antigen, whatever you want to precipitate or separate. That is the main thing. But in your laboratory, you don't have that antibody. So in that case, what you have to do to circumvent this problem, proteins can be tagged with an epitope to which a high affinity antibody is available. What is that means? If you consider key, this is the protein, okay? You want you don't have antibody against this protein, but you just attach one probe or attach one separate epitope tag. So that tag will act as the epitope. Epitope means epitope is actually recognized by the antibody, है ना, which is present on the surface of antigen. So if you consider that whole antigen is not acting as the epitope, only the tagged part is acting as the epitope, the small part. Okay. So in that case, in this way, if you are precipitating any antigen, that is called the tagged protein immunoprecipitation. Okay. So now if you consider what is happening actually, this is the actual process. In this case, directly in the protein, you are not adding any epitope protein. From the starting point, that is from the gene level, you have to add. Like initially, if you consider this is the protein of interest, this red color, I'm adding red color. Okay, so this is the protein of interest, but you don't have antibody against this protein. So inside the with the gene, you just add tag protein. Okay, one tag sequence. Here I'm giving you green color. So you just add tag sequence. So what will happen? Together with the protein of interest, this tag protein will be transcribed and translation will be there. Okay, now you have this protein of interest together with that tag protein expression. So in this figure it is given ना एक green color का part है, that is the protein of interest. With green color part, some blue color part is there. The blue color part is actually your epitopic part, whatever you tagged. Now against this tag you have antibody. If you add antibody with beads, what will happen? This whole complex will be precipitated with the antibody. Okay, so antigen is actually, or antigen of interest is actually precipitated with the epitope which is added before the precipitation. Clear? So this is nothing, you just making artificially one tag and then you are doing the precipitation, nothing else. Whole antigen is not antibody is not developed against the full antigen of interest. Clear beta?
हाँ
है ना? So we have discussed few methods related to immunoprecipitation. One, first here it is tag protein immunoprecipitation, then a REAP, that is the RNA immunoprecipitation, then what we have discussed before that,
chip हाँ
chip, yes, chip DNA precipitation. Then we have discussed it is co-immunoprecipitation, okay, and normal individual immunoprecipitation, individual protein immunoprecipitation. Okay, so DNA interaction, DNA protein interaction we have done through which method?
chip
DNA protein,
chip हाँ
chip, yes, and protein protein interaction,
go
okay, RNA protein interaction.
yes
Yes. Or if antibody is not available, you can, if you can do the tagging. So in that case, tag immunoprecipitation. Clear? I hope clear. So I'm just crossing it. I hope there will be no problem. Now the connection is stable, na?
yes
After
ma'am
this discussion. Okay. Okay. Chalo. For today, we can leave. Okay. Bye. Tata. Next day.
please send the PPT of yesterday
Ha
also
ha 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 ha. Yes, yes, yes. Western blotting also. I'm sending the PPT if you don't understand it. Okay. The next day I will discuss it. Okay. You just let me know, okay, whether you understood or not. I'm sharing one PPT regarding Western blot, Northern blot, Southern blot. Very simple method.
and
Very
PPT
simple method.
before that one also ma'am for plan
That is also I will share. That is also I will share. Okay. Chalo. Chalo. Bye
thank
bye
you ma'am
bye. Take care. Bye.